everyone. Welcome to another episode of Drug Development Diaries. In this series of different episodes, we are discussing a book called Approved: The Life Cycle of Drug Development. This book that was recently released by Springer, we have been talking about the overall process of drug development, from conceptualization to bringing the drug to the market, about different challenges and pitfalls and exciting experiences. Have different chapters, and in each episode of this series we are inviting authors of these chapters and we are having a candid conversation with them about their experience of writing this chapter, about how they have structured this chapter, and about what exactly are the expertise that they are bringing in front of us. So on similar lines, in today's episode we have Dr. Sanjay Khare and Dr. Taruna Arora, who have authored a chapter on biologics. uh, and also, as every other episode, we have Dr. Naren Chirmule, who is a co-host, also here on this episode. So I would request Naren sir to take it up from here, introduce our guests, and get the episode starting. So
Okay.
welcome everyone. Let's start the episode.
Thank you, Vihang, and thank you for managing all these episodes that you've been doing. Thank you very much. Welcome, Sanjay and Taruna. I met Sanjay and Taruna maybe 25 years ago, 20 years ago at least, when we were working together at Amgen in Thousand Oaks, and I think we connected immediately because of the common interest of biologic drugs that we were all working on together at that time. And I think we were working on things like Embril and other drugs like that. And I think the field was 25 years ago. It was a beginning to take off. Just a few drugs had been approved. I think the benefit of biologics had not yet been established in the community. And so I think both Sanjay and Taruna have enormous experience in both manufacturing and the entire process of drug development. Both of them have PhDs from in their respective institutes. So Taruna Arora, who has a lot of experience in early discovery to bringing the lead candidates to development. I think that's a lot of experience in that area, and she'll speak a little bit about it, and a lot of it is written in the textbook. Sanjay has a very interesting journey. In addition to working in companies like Amgen and others, he also had an entrepreneurial journey of his own, and he can maybe speak a little bit about that. So with that introduction, welcome to the podcast, Sanjay and Taruna.
Thank you. It's really a pleasure to be here and great to see you both again, Bihang and Naren. Yeah.
Thank you. So let's start with a very basic question about biologics. So maybe you can explain what is biologics and how they are different from like say small molecule drugs.
Yeah, so biologics are the medicines in a simple way said. They are medicines that are produced in living organisms, or they are a component of the living cells or living organisms. So these include like vaccines, proteins, hormones, enzymes, and of course, the antibodies, as well as now we have next generation of antibodies, multi-specific antibodies, and even CAR Ts, I would say, are a derivative or another version of biologics, cell therapy though. So biologics differ from small molecules in many ways. First, starting with small molecules, generally they are taken orally as a pill. They have mostly, short half-life, cleared very fast. Biologics, because of their structure, tend to be more complex. They can have post-translational modifications when they are produced from living organisms. So they're larger in size. They can be larger from anywhere from 100 to 1,000 times larger from small molecules. So in addition to product complexity, they also are generally administered as either intravenous or subcutaneously. Although now there are some efforts going on to make them also orally available in a nanobody form.
Yeah.
So those are some of the differences. Biologics being more complex definitely require more process development, optimization for the goal of bringing more efficacious and safe and high quality drugs to the patients, because in the end, patient safety and efficacy matters the most.
That's very good.
So those are, some of the things I can think of
Good.
as a difference in small molecule and large molecules. Sanjay,
Yeah,
you have to.
Sanjay.
Yeah, I will add couple of things. Uh, yeah, number one that uh, biologics do differ from small molecule in another way that uh, in small molecules uh, you don't have problem with the anti-drug antibodies, so there is no neutralizing effect there. Drug is there for a short period of time. That's another thing. So let me just expand on these two. Um, First, because they don't have anti-drug antibody in small molecule, uh, there are as long as drug is there in circulation, it is going to be effective. However, in in terms of in case of biologics, if patients develop anti-drug antibodies, then they may neutralize the effect of drug, and therefore they may not be effective.
Mm. Mm.
Second one is that, of course, Taruna pointed out the long-lasting effect in the body.
Yeah.
It is good and it is bad. The good is as long as it is doing good thing, but bad is that effect in the body is going to last for much longer period of time compared to small molecules. So yes, we all love biologics, we all love antibodies, but we just have to be a little more careful.
Very good. Can you give some examples of biologic drugs that are there in the market today?
Yeah, I think some of the ones which have been very transformational. First one that comes to mind, obviously, is insulin. Insulin is an hexamer, inactive before it is secreted. And initially, it was being used as an extract from animals' pancreas, and that's how it was being used as a drug to treat diabetes. Still, huge, huge step in terms of meeting the unmet need for patients suffering with the type 2 diabetes. And then with the evolution of analytical techniques, later on with the recombinant DNA technology, now you can produce the insulin as more, homogeneous product, which can be monitored analytically for quality. And again, it's one of the, I think everyone knows this drug very well. When it was being produced from animal pancreas, there was always this issue of the quality, aggregation, immunogenicity, which was resulting in reduction of its efficacy as well as also some of the safety issues. But
Mm.
with the advent of recombinant DNA technology and production in a more consistent manner, it definitely has helped to control some of these adverse events.
Yeah.
The other example which comes to mind, and maybe Sanjay can talk about this more, anti-TNF drugs,
Mm.
uh, Enbrel and also Humira, right?
Mm.
So Humira is now like a 30 billion drug, and is used for the treatment of rheumatoid arthritis.
Mm.
But there have been immunogenicity issues as well with,
Mm.
especially with Humira. So I don't know, Sanjay, if you want to speak to any of these drugs since you have worked on both.
Yeah, I think before I discuss those, I would briefly like to give the evolution of biologics because that has been actually very interesting journey from where we started. And Tarna, thanks for bringing up this story of insulin. You know that has been helping millions and probably billions of patients now. But the journey has not been very easy with biologics. I can tell you that. I mean, right from the beginning, when the new molecules were, the recombinant technologies were produced, some of them became really successful drugs. And those were body's own proteins, such as erythropoietin or GCSF or granulocyte stimulating factor. But there has also been a story where these bodies' self protein, when they were produced recombinantly, basically did not work in patients. And they did not work primarily because there were anti-drug antibodies were formed. I mean, big example that come to my mind are two of them, and those projects were started at the Premier Biotech Institute, Genentech. One of them was MGTF program for stimulating platelets, and every single patient who got that drug, they developed anti-MGTF antibody, hence leading to depletion of platelets instead of stimulating platelets. Of course, that the part of the problem was also seen with erythropoietin when a competitor of Amgen was trying to set up their own shop to produce erythropoietin. The other example is anti-TNF drug, TNF receptor R1. Uh, of course, I'll discuss about this TNF story, but it's really, really interesting. And maybe I can begin TNF story right from here. So TNF being one of the critical components for the body, especially for the inflammation. A lot of companies jumped into the ideas in late, well, late mid to late 80s, and they were all trying to make anti-TNF product, anti-TNF biologic. So there were companies that were using a receptor fusion protein, so TNF receptor 1 that will soak TNF. At the same time, company like Immunex were working on TNF receptor 2 fusion protein. And TNF receptor 2 fusion protein from Enbrel, which is Enbrel, became a product because there was not much immunogenicity. However, TNF receptor 1, it has 4 domains, and seems like patients who were getting this TNF receptor 1 fusion protein, they were all getting fair amount of those patients were getting anti-drug antibodies. We had a phenomenal, great scientist in Amgen those days, and he did real Japanese fellow Hiko Kono. He did a good job in truncating that molecule, so that molecule is still effective the way it should be, but it lacks immunogenicity part. So your
Less
patient won't get
active.
immunogenicity. Yeah, and certainly, with his research, Amgen did put that molecule in patients. Very few patients had immunogenicity, or there was a good tolerability of this molecule. And he has to truncate that to 2.6 domain instead of 4, and that provided that that basically got rid of immunogenicity issue, but it was still efficacious.
Yeah, so Sanjay, I'll ask you a question related to this story that you're telling. Looks like the journey of biologics has not been a smooth one. So, it's been 25 years later. What are the current trends of biologic drugs that are being developed now?
Yeah, good question. So now I would say one of the biologic that has become awfully uh, interesting for all protein scientists is antibody and antibody derivatives. Okay. When I recall my early days working on the antibody back in my days in Amgen, we were basically wanted to stay away from antibodies because antibodies being a foreign protein, one could make anti-drug antibody or anti-antibody.
And
Yeah,
even effector functions. Nobody wanted to have a cytotoxic function associated with antibody at that
Yes.
time. We were working with IgG2s, right? I still remember that. But since then, right, we have seen how much field has evolved, not just using monospecific antibodies, which may have multiple functions through both variable regions, targeted target binding region and FC. We are now working with bispecifics, we're working with antibody drug conjugates, T cell engagers, right? And a lot of these initiatives, I feel fortunate to have seen, when we were at Amgen working on these innovative biologics. Field has matured now. Certainly, the goal has become to be able to have more efficacy while keeping them simpler, but also, multifunctional.
Yeah.
So,
Yeah.
and so that's for the modalities in matching to mechanism of action, making them more and more superior in efficacy. But the other is discovery, bringing these faster to, and that's where now everybody knows this term AI/ML. So it feels like we were doing that at some scale, but now it's definitely a much larger scale, and you are incorporating AI/ML driven approaches in discovery, and then in process optimizations, manufacturing, as well as including clinical development. Sanjay, you worked on some of those with a company. Maybe you can speak more to the AI/ML part.
Yeah.
Well, I would say those were early days of artificial intelligence, machine learning. Actually, I reached out to uh, Google when Google just acquired this AlphaFold protein discovery platform. And in the whole idea was, how one could make use of those structures and maybe design antibodies instead of generating those antibodies in transgenic animals or you know different mice, rats, or rabbits. Um, so there were two primary approaches were taken, and I think that the field is still evolving. But let me tell you, in the beginning, from the AI/ML, there were two primary approaches taken. Number one, people pulled out the sequence, and then the sequence, the linear sequence, they will design the antibody CDRs based on those linear sequences of the protein. And the other company took the approach where they will have this conformational epitope, and and then they will design antibody based on conformational epitope. In my mind, when you are developing these molecules, we are looking for three biologies. Okay, particularly in case of antibodies. Okay, number one, you are looking for inhibition of the target. So you're blocking. Those are called blocking antibodies. Second one is agonistic antibodies, so you can mimic the ligand, so you can activate the receptor, that is agonistic antibody. And third one is just an anchor. You don't want it to agonize or to block any molecule, but just to anchor there to get a targeted delivery of another molecule or cytotoxic drug or anything that you could relate to, or the cells in CAR T for that reason. So I think these are the three principles still maturing. We still yet to see whether a AI machine learning driven sequences go in patients, unless Taruna knows something that is already in the clinic.
No, not yet. Not from the discovery, but certainly there have been applications of AI/ML in manufacturing and also for the clinical operations, data management, for sure.
Yeah, so very exciting times to be in the field of biologics. So, but let me hand it over to Vihang to ask you a few questions regarding his take on the text on the chapter.
Thank you, sir. This is a wonderful discussion, Sanjay sir and Taruna ma'am. Very exciting to be here on a part of this podcast. So let's divert ourselves to the chapter that we have written here in the book, Approved: The Life Cycle for Drug Development. So could you share a bit insights on how this chapter is structured? What should the reader look forward to? How they should navigate through different sections and make the best use of the content that is presented in the chapter?
Yeah. So writing this chapter was very, very exciting. Generally, I have been more in the role of, having my team members, right, and you're reviewing and getting back into that mode was also very interesting. But biologics, as we have been discussing, they're so complex. There's so many varieties of modalities, and how you bring it together in a chapter in a concise way, that was very important, so we discussed, and we felt that it would be good to classify them into, the proteins, endogenous protein-based medicine, smaller size versus FC-based proteins, recombinant proteins, and then antibodies and next generation biologics. Because I think this is where what the current trend is. So I would like our readers to really understand these different classes of biologic modalities, why they were designed in a way, what was the criticality, what has been the historic journey to get to, monoclonal antibodies and now to next generation biologics, how this field has evolved. So those are, I think, which is very important, right? We definitely are seeing now more and more complex biologics being developed and also approved. So, and hopefully, readers will enjoy that journey from starting with the history and different classes of modalities, and then the future trends. Yeah.
Great, great, great. Would you like to share a bit about how was your writing experience? Because a very long time of your career you have spent in the industry and also in academia, but it's more have been a research-centric endeavor. But writing something for something young professionals and for students is slightly different than what you do on a daily basis. So how did you feel getting back to the desk with a pen and a paper and going through multiple rounds of editorial revisions? How was it? And would you like to say something about it?
Yeah, first of all, thank you to both of you and Naren for including us in, as the authors. And I told you separately, that it was very, very a great, satisfactory experience. Long it was because you're trying to put together everything you've learned, and you also want to make sure you are not missing. So you start with the outline. This is what, we're going to work on with the different outline, and then filling, content for those outline. We did have our differences in opinion, Sanjay and I, because we come with different
Right,
perspective. But also, I think it helped us to also complement, our sections on, like from his point, the drugs that are approved and also safety aspects, which are important. And for me, I was thinking more, design goals and how you want to design a drug to match with the intended outcome for therapeutic. So yeah, and I think along the way you have helped a lot, Vihang, and there were others who were also helping with the editing. So yeah, I enjoyed it a lot, Sanjay.
yeah, sir.
Yes. Well, you know, this is one way I also learned how to be a better listener. But besides that, um, I think it was very complimentary, and this is the first chapter, actually, first chapter that we have written, or any publication Taruna and I have written together.
Wow.
So, it was interesting, and lot of things we talk in a day-to-day life, but writing something together where you have differences and you know not differences about the science that we are talking about, but how it should be written.
Hmm.
You know, and I am kind of one of those person like who likes evolution, and whether it is antibody evolution, then we started with the mouse antibody, made chimeric, and then humanized antibody, and human antibody, and then multiple variant of the antibody platform that we're using. So that was an interesting journey. um, just to get into all that literature, even though when but when you're writing, you have to be very systematic in putting everything together. So I would say I really enjoyed educating myself and putting this together in this book. And again, you know, thanks, Naren, for the opportunity. Really
Well,
appreciate this.
no,
Uh,
thank you both for writing. And I must say that me included, I feel very grateful that we wrote this pre-Chat GPT time.
yes, well, I would credit ChatGPT as well, because
Okay.
certain things that you get to know right at that moment,
Yes, yes,
it
yes.
can help,
Mm,
but you have
can
to
help.
cross check. You
Yeah,
have to cross check,
absolutely.
because a lot of times, I was pulling the information and that was incorrect in ChatGPT, because ChatGPT is still being, getting educated, I
Evolution.
would say.
Right,
Yeah,
right.
yeah,
Right.
Agree, agree. Yeah,
Absolutely.
yeah.
So finally, what advice would you like to give it to students or young professionals, considering you have spent so many years in this field? If there is a student who wants to have their career in drug development, what is it that the student should focus on? What skills that they should develop, and what is it that they should keep in mind while walking this path?
Taruna, you want to start?
Yeah, I think the whoever wants to be in the field of biologics should be asking questions what I'm doing, how I'm doing, and why I'm doing. Because biologics that we work are not just simply a technology purpose. We are developing them to meet the need for patients' benefit. So it's very important also in my mind to marry the biology and the modality. So you need to have some understanding of biology principles so that you can design appropriate modalities to fit with it. Be open-minded. Do not be just, whatever you may have learned during your master's or PhD is not the only way probably to do the things. You need to be open-minded to learn new things, broaden your horizon, and flexibility, adaptability is also very important. Understanding of biochemistry, especially in biologics, we work do a lot of protein engineering, right? So having some fundamental knowledge of biochemistry, amino acids is important. But but always stay curious, ask questions. There's no dumb question, and continue to broaden your horizon. Me myself, I came from immunology background and became a member of protein science and built my career in biologics. Yeah,
I know.
Sanjay.
And Sanjay, before you start, maybe what I can ask you to do a little bit, if I can interrupt for a second,
Sure.
maybe if you can give your experiences, but also maybe talk about your entrepreneurial journey and how younger folks could think about entrepreneurship in this area.
I would agree what Taruna just said. You have to be open-minded, and the newer technologies are definitely going to help, but experience, in-person experience is very important. And then you probably need to talk to people who have gone through the drug development cycle, and they have seen both ups and downs of drug development. I mean, having said that, 90% of drugs fail in the clinical trial,
Right.
and for a reason, because our body is very complex. No matter how you are developing, you may be coming out with the best, you may be using best technology, you may be developing a best molecule, but you would still have some unanswered question why this drug fail. Some fails because you have a safety for, because we don't understand the biology, how these molecules are interconnected. And sometimes it fails because of uh, reasons that none of us understand. We all thought that our body is immune tolerant to self protein, but then when we started to see uh, anti-drug antibody against self protein, it became challenging. Okay. I would say, one thing that everyone has to keep in mind that, how what you learn from your institute or from your educational background or some experience in the company, you want to continue doing that, or maybe there are some ideas that come to your mind, and it may not be fully mature idea, but you may want to explore that, and in the resources or the timing may not be the best with the environment that you are growing. And that is one of the things that actually came in my path when I was working in Amgen, and we were, that is back in like 2007, 8, or maybe even a couple of years earlier. The idea was like, okay, anti-TNF drugs do phenomenal job for rheumatoid arthritis patients and psoriatic arthritis psoriasis patients, but it's still benefit to those patients. Majority of those patients is about 50%. It means 50% of disease complexity is uh, you are correcting, but there may be some additional mechanisms involved, and how you can take care of additional mechanism. And we started working on those in animals. We were able to actually cure lupus-like disease, and we establish lupus-like disease uh, in rheumatoid arthritis models. Also, we were able to see some of that effect. And then we were developing biologics for further advancement. But and that is when and that was one part of the story. But the other part of the story, that a lot of these cytokines, those are being used um, as biologics for cancer treatment, or for some infectious diseases, such as interferon being one of them. And the whole idea was that every patient who takes interferon, they get antiviral effect, or they get anti-cancer effect. But it is a horrible therapy. You patients get flu-like symptoms, fever, and number of other things. So to overcome that, we were thinking about, okay, can we make a biologic that is more targeted? And based on that, actually, it didn't work in that time in the company situation where I was, but I started a company called Immune, and right from the drawing board, with the help of some key key collaborators such as Sherry Morrison, and many other company executives, um, took this molecule from drawing board all the way to phase 1, phase 2 clinical trial. And we proved actually the modified targeted interferon, so antibody fused with interferon, was working like a prodrug. Interferon was active only when antibody was binding to the target. And that is the next generation of things. Is still evolving, I would say. If you see antibody drug conjugates, primarily the same thing. You don't want the cytotoxic drugs to be working everywhere in the body, so you want to control their effect in the targeted tissue. And same thing people are trying to do with several different kind of biologics. So you have some idea, you need to explore that idea. A lot of the drugs are made not for the um, uh, intended benefit for patients. It was serendipitous finding, and you took the path for those serendipitous finding, and you make the drug. I was giving example of anti-TNF or even Viagra. They were not started for that purpose in the beginning. So you have to be open-minded. You have to be open-minded, and of course, uh, and of course, you need some support.
Yeah. So Vihang, back to you.
Amazing, sir. These anecdotes and these experiences are quite insightful. I'm sure our viewers and our prospective readers will definitely find this discussion very useful and insightful and inspiring as well. Right. So we are coming to the end of this discussion, sir. Naren sir, I request you to go ahead with the closing remarks.
Yeah, so thank you, Sanjay and Taruna, for coming to this podcast. I think one of the reasons we are doing this podcast is not only for sharing the information that you have just shared, but also put a face to the name sometimes. You know, when people are reading the book, they can actually see the person. Sometimes it also makes a difference in how you read the book. So with that, thank you very much for this episode. Exciting discussions, and I think these discussions will, I'm sure, continue. So many more discussions to have.
And it
So
probably reminds you of all the collaborations we had at Amgen.
exactly, I don't feel like stopping this podcast. There are so many more questions that you have to do, but anyway. So thank you again for coming to this session, and
Thank you.
yeah, yeah.
Thank you, Naren. We should start the next one now.
We should. I think we will very soon. Yeah.
Okay.
Okay. Yeah.
Absolutely.
Thank you,
Great,
Dion.
great. Thank you. Thank you, sir. Thank you for the very inspiring episode. Thank you to all the viewers for watching this episode. Uh, Definitely go ahead, grab this book, read the different chapters that are written over there. Tell us what you think about it. If you have any questions about the different concepts presented over here, do let us know in the comments and have an interaction. Let us know what else would you like to learn about these topics and benefit from the discussion that we are having over in this podcast. So until we'll see you in the next episode. Until then, stay curious, stay inspired, stay tuned with Drug Development Diaries, and see you in the next episode. Bye bye.